much, Perry. Thank you very much, Perry. I'd like to echo those words of thanks to the committee who put this on and made it possible for us to hold our symposium this year. So title is Serendipity in the Discovery of British Columbia's first KPG boundary section, a snapshot of marine phytoplankton turnover and paleoenvironmental change in the North Pacific realm. My supervisor at the University of Victoria has been Vera Pospolova, who's now with us from University in Minneapolis, Minnesota. So I'm going to start by introducing dinoflagellates. Dinoflagellates are single-celled protists, which is Latin for whirling whip because of the flagellum that encircles them. They're found in all oceans from the Triassic. Some are freshwater and some live in lakes, as well as the ocean. They are the second largest phytoplankton group, the first largest being diatoms. They are auto, hetero, and mixotrophic. And most importantly for my work, they form a resting cyst stage or hypnozygote, also known as a dinocyst for short, comprised of a carbohydrate-based biopolymer called dinosporins. They are highly abundant. They can be found in just 1 gram of sediment with thousands of them. They're widely dispersed, easily and well preserved, easily collected. You only need small samples. And many groups and species are ecologically, latitudinally, environmentally, and temporally constrained. Furthermore, dinocysts are tools for paleoenvironmental reconstructions and relative dating. In deep time studies, they provide insights into paleoecology, so the relation to other fossil assemblages, paleooceanography, ocean circulation patterns, paleobiogeography, spatial range and provincialism, extinction and succession, so temporal ranges and diachronaity, paleoproductivity, so nutrient input and upwelling indicators, as well as paleoclimate, so latitudinal temperature gradient indicators as well. This is a complex slide, but it's just to illustrate that dinocysts have preferential habitats. Some like to be more inner shelf, more coastal, estuarine, more tolerant of brackish waters, whereas others provide more of an offshore signal with potentially oceanic input and coastal upwelling from deeper waters. Motivations for this work are that Cretaceous Earliopaleogene research is scarce in the North Pacific. We only had some work done in California and in Japan. Dinocysts are, of course, indicators of responsivity to the KPG event if they're environmental indicators. And the North Pacific is the last vast geographic frontier for deep time marine palynology, especially for the KPG boundary interval. So we're all probably familiar with the end of the Cretaceous mass extinction at approximately 66 million years ago that wiped out a large portion of the fauna and flora. Because we had a bolide impact coinciding with Deccan volcanism and those major flora and faunal extinctions, global warming of approximately 5 degrees over the 100,000 years ensuing, and some short-term cooling, as well as primary productivity recovering over that 500,000 years later. That's a snapshot of the KPG boundary at the global stratotype section and point in El Kefa, Tunisia, showing that classic boundary clay layer. For this research objectives, we had classification and taxonomy, which gave us the opportunity to survey and describe the taxa present and ID the index species, relative dating, constructing the biostratigraphy across the KPG boundary. And for paleoenvironmental reconstruction, we could assess the depositional setting, the primary productivity, the paleoenvironmental reconstructions locally and abroad, as well as phytoplankton paleoecology and changes across the KPG boundary, and to correlate the North Pacific intervals with other regions and to synthesize these findings with a global data set. So our region here on Vancouver Island, and then just south of Campbell River is the area of excitement. That is the Oyster Bay Formation, yet to be formally described, which is unconformably overlying the Nanaimo group strata. Here's a strat column of the full Nanaimo group, and there we would be right above that with the Oyster Bay strata. And it's broken into 3 principal outcrops. A there is classic Appian Way. B just to the north is what's been deemed the Linwood section, and C to the south is a small outcrop along the Oyster River. And this is a bit of a schematic diagram, aerial view of these locations and their outcrops. Appian Way along the coast, B, the Linwood, and C, Oyster River, with all those red dots indicating sites of matrix sampled for this study. So standing in the Linwood, looking to the northeast, we have some massive sandstones and alternating mudstones. The Appian Way there, a little quick video looking from the southeast. We're panning to the northwest, looking up toward the Linwood locality there. Shows you those alternating beds of finer and coarse grained strata. And standing on the syncline axis there at Appian Way, looking to the southeast and panning up to the northwest. And then on the Oyster River, a small outcrop of just a few meters, but nevertheless, the same strata, sandstones, and mudstones. So the serendipity aspect of this study on this slide has to do with the basal Linwood. So in the pilot part of this study, I went to the Linwood locality, took 3 samples, one at the top, one in the middle, and one at the bottom. And the one at the bottom came out of this notch in the rock right about there. If you can see my pseudo laser pointer right about in there, we got this little depression. It wasn't quite a concretion, but it was semi-consolidated. So I thought, okay, there's a candidate for a sample. And then lo and behold, it was absolutely loaded with Palnodinium growliter, which is a species that is perhaps the greatest indicator for the upper Maastrichtian in the Northern Hemisphere in terms of palynological assemblages. And it wasn't just that it was Palnodinium growliter present, it was a Palnodinium growliter acme, which was constrained in the section to about 10 centimeters. And given the thickness of the whole section, which was the Linwood section, which was about 48 meters, I figured that the probability of me sampling that one on the first whack was about 1 in 650,000. So there you go. It was enough to really give you a kind of kick in the pants to get going and get excited about this project, given that there's definitively Cretaceous material. Well, boundaries that has to be there somewhere, and it certainly was. So the matrix was sampled in baggies and then brought to the lab, digested with hydrochloric acid and then hydrofluoric acid to remove the carbonates, and then the siliceous materials to give us the final organics that we then turned into smear slides after introducing Lycopodium spores, which were the modern moss spore used as a numeric control, so that we could then determine absolute and relative abundances for inferences based on ratios. And there's a smear slide, for example. It was made from material that was sieved above 50 microns, but below 120 for light transmitted microscopy, and also epifluorescence microscopy, which you see there, the illumination of specimens revealing to us a proxy for autotrophic capabilities, meaning that these guys made their own food and didn't just hunt if they illuminated. So 2 key index taxa here, of course, to the right, Palnodinium growliter, as mentioned, for the upper Maastrichtian, and then Dania californica for the basal Danian up section. So this is an example of the diversity that we have here. These are heterotrophic dinoflagellates with classic kind of what's called a peridinioid morphology with their angular horns. And then we can see that some of these varieties also illuminate, telling us that they had mixotrophic capability and that they weren't just hunters. Some more diversity here showcased by a whole myriad of different forms, different fibrous processes, and the like. They all have what's called a characteristic opening, known as an archipyle, which I'm sure you'll notice on some of these. That's where the cell hatched out and went about its life. We'll get that. There we are. So we've got little openings there, openings there, openings there, and so forth. A laser pointer working there. And then some have more elaborate structures where they're all very spinous, very branching, very intricate, for different means to enable their suspension in the water column for a longer period of time, because if you sink and you hatch out, you have a lot longer of a distance to swim back up to your favorite position in the water column. This slide summarizes pretty much everything in terms of the assemblages that were recovered. We have the three sections on the left there, Linwood, Appian Way, and the Oyster River. And these signals, or these patterns, are the different groups of dinoflagellates and terrestrial spores and pollen, indicating the turnover event that happened there post-Cretaceous. So we have an expanded KPG boundary interval that is essentially at the uppermost part of the Linwood section, where we have a major turnover in the assemblages, moving from a more offshore signal to a more inshore signal, with influences from terrigenous outflow and brackish waters. This is an exploded view of the bottom of the Linwood, just to show you there that in the yellow, that is this spike of the Palindinium growler as they peaked right at the late Maastrichtian. So synthesis of the overall global data set was performed as part of this study as well. So all the world's literature, 676 reports collected from 435 published studies, show us that 87% of the reports of the 10 key index species in the Oyster Bay Formation are constrained within 6 million years crossing the KPG boundary. And these are some of those selected taxa plotted of their positions all across the world in relation to where we are up there with that little star. And then also for some of the more predominantly heterotrophic or peridinioid dinoflagellates. Now, also, there aren't only dinoflagellates, of course, in the palynological assemblage. There are a myriad of other things. We have pyritized endocasts of silicaflagellates and diatoms on the upper right there, scolecodonts in the top, the CDE, which are the mouth parts of marine polychaete worms, acritarchs, and sporomorphs, palomorphs of all different affinities, as well as the internal organic linings of foraminifers, so K and L down there in the bottom left. But most of all, the second greatest diversity is, of course, with the terrestrial spores and pollen, which would be the next jumping off point for future research. So the highlights of this study are that the Oyster Bay Formation revealed the first KPG boundary interval west of the Rockies based on biostratigraphic controls, and potentially the oldest Paleogene section in coastal BC. And the Appian Way is definitively Danian. BC fossil or dinocysts record the phytoplankton and paleoecological turnover across the KPG boundary, which is consistent with other coeval deposits across both hemispheres. And the microfossil assemblages record signals of coastal to estuarine paleoenvironmental fluctuation over 4.5 to 5 million years. And also, what greater reinforcing piece of evidence for terrestrial input than the incredible plethora of plants that have been described by Ruth Stocky and her group over the last 20 years. Dinocysts also provide signals that indicate periods of localized elevated primary productivity, likely owing to upwelling of nutrient-rich waters and that terrigenous outflow, as well as convergence of boreal and subtropical taxa, indicating depositional settings that are probable with their paleolatitude near the present location. And the Dinocyst taxa of West African affinity suggests that warming surface waters into the earliest Danian carried from an east-west transatlantic current those dinocysts over to our west coast and then up north the west coast of North America. I'd like to thank everyone who made this possible. So much of this was possible only because of the efforts of other people lending their skills, time, and expertise. And with that, I'm happy to take any questions you may have.